My name is Tom Worthington. I'm a researcher in the Conservation Science Group and part of the CCI committee that organises these seminars. This is the last seminar before the Easter break, and then we'll be returning in about 4 weeks' time. So today's speaker is Dr. Michelle Jackson from the University of Oxford. It's my great pleasure to introduce her on this really interesting topic today. So Michelle completed her PhD at Queen Mary University in London in 2012 and has worked as a postdoctoral researcher for 3 different universities, both in the UK, Bournemouth and Imperial, but also the University of Pretoria in South Africa. She joined the University of Oxford in September 2019 as an associate professor of freshwater ecology. Michelle's research aims to understand, predict, and manage the effects of anthropogenic stressors, such as climate change, species invasions, and pollutions on aquatic communities. And she's particularly interested in the food web mediated effects of multiple interacting stressors, in both streams and rivers. She is the secretary of the British Ecological Society Aquatic Group and an honorary researcher at the Centre for Invasion Biology at Stellenbosch University. This is sure to be a really, really interesting presentation, so I'm going to pass over to Michelle now. Thank you.
Thank you, Tom, for that really nice introduction. I'm just trying to share my screen. Can you see that now?
Yeah, that's great.
Okay, thank you very much. So as Tom said, I'm going to be talking about variation in the responses of aquatic communities to climate change. So there's really 3 parts of my talk. I want to start off talking about warming in streams in Arctic locations before moving on to talk about more rapid warming in experimental mesocosms. So this is some work from the UK. And then I'm going to finish up by talking about how climate change might interact with other anthropogenic stressors. Sorry, I think it's moved slides now. I got kind of caught up in Zoom there. So first of all, I want to talk about why I am interested in freshwaters in particular and why these are the ecosystems that I research. And the first reason is that freshwaters are rare. So if we look at all of the water on Earth, much of it is salty, about 97% is in the ocean. And of the remaining 3% of water that is fresh, most of that is held up in groundwater, in glaciers, ice caps, ground ice and permafrost. And actually only 0.8% of the Earth's volume of water is available in lakes and rivers and wetlands as habitat. And if we look at this as a surface area, you can see that we're very much a blue planet. So this is the surface area of Earth. And that large dark blue part is the ocean. So about 70% of the Earth is covered by the ocean, with most of the remaining 30% in green and the red and orange colours there covered by land. And it's only that small sliver in the middle in light blue that is the Earth's surface that's covered by fresh water. So they're very rare habitats. But they're also disproportionately biodiverse. So if we look at any taxonomic group from diatoms through to macroinvertebrates, fish and mammals, they have very high biodiversity in freshwater ecosystems. And a good example of this is fish in many lakes. So the example at the bottom there is Lake Malawi, which has between 700 and 1,000 different species of fish. And overall on Earth, of all of the described fish species, 55% of them are freshwater or rely on freshwaters for part of their life cycle. So for instance, salmon. And this is quite striking. If we go back to this figure here, it's just in that small sliver of fresh water that we get all of this biodiversity. And we see similar facts if you look at other taxonomic groups. For instance, 25% of all described mollusks are from freshwater habitats. The third reason why I think freshwaters are a really important area of research is that we really need them. So they provide many ecosystem services. So these are the benefits that humans get from the natural environment. So really apparent one from freshwaters is, of course, drinking water, but also things like fish protein and less obvious services such as flood mitigation or nutrient cycling. And finally, freshwaters are, in my opinion, one of the most threatened ecosystems on the planet. So this is the WWF Living Planet report, which looks at population declines in vertebrate species since 1970. So on the y-axis, we have a kind of baseline value of population abundance of 1 in 1970, and any deviations from this show either increase if it goes up or a decrease if it goes down. And this is the global trend, so looking in multiple populations across the globe. And for the freshwater part of the database, this is about 4,000 populations covering 1,000 species. And on average, these populations have declined by about 84% in the last 50 years or so, which is quite a scary number. And that's compared to about 40% in terrestrial and marine ecosystems. However, it's important to note that it might be a small number of populations with severe declines that are driving these trends. So often these large-scale analyses looking at global trends can obscure variation at more local scales. So this is a re-analysis by Lung et al., which looked at the same living planet data. So across all of the ecosystems, so about 15,000 populations, and found that if they were to remove a few populations from the database, that we get actually a much more positive outlook. So there's a few populations which are declining very severely, and others might be much more stable. And so they propose this different way of looking at the Living Planet Index, where we consider taxonomic groups and location rather than looking at one overall global trend. So here you can see in yellow and green the populations which are staying more or less stable over time. In blue, there's some that are actually increasing, and it's only really the populations in orange and red that have got these negative trends and are driving these headline grabbing declines. So it's important that we look at this variation to get a real idea of what's going on. And the reason I mentioned this is because this is something we had to consider in our research looking at warming in Arctic freshwater ecosystems. So this is the what we call the Ring of Fire project, which I worked on when I was a postdoc at Imperial College in London. It's led by Professor Guy Woodward. And here we have 5 circumpolar locations. And the reason it's called the Ring of Fire is because they're all geothermal locations. So we were interested in how warming in these regions will alter biodiversity and whether it changes between the regions. So is there an overall trend, and is that trend apparent in all 5 locations? In this project, we're also looking at the impact of warming in various different scales. So we're interested in gene to ecosystem responses, looking at microbial communities constructed in the lab, also using mesocosms, which I'll talk about a little bit later. But I want to start off with talking about our geothermal systems in the Arctic. So in each of these locations, so we have Alaska in the USA, Kamchatka in Russia, Svalbard, Iceland, and Disco Island in Greenland, we have what we refer to as a natural laboratory. So the figure on the right there shows one of these in Iceland. So each line represents a stream, and then the circle with the temperature inside shows the temperature of that stream. So the streams vary in temperature because of underlying geothermal activity. And you can see here in Iceland, our temperature ranges from about 5 to 25 degrees Celsius. And these streams are very close together. So this is a drone shot as an example of that, showing 2 streams. This one here is highlighted in orange on the map as well, is 25 degrees. And this one, which is just a few meters away, is 8 degrees. So they're so close you could almost have one hand in one and one hand in the other. And they're really similar in every way except for temperature. So we can isolate temperature as a master variable and see how it affects everything from the microbial community through to the whole food web and the ecosystem processes that food web drives. So we would expect the communities in these regions to be shaped by these environmental conditions. So firstly, the location, which is why it's important to look at this across these different locations, because the geography and geology of that region will affect the regional species pool. And then the most important filter that we're considering is the environmental filter of temperature. And we expect that to alter the community structure and ecosystem processes. So this stream network that we have in these 5 locations allows us to look at the effects of climate change in these small spaces rather than having to use a latitudinal gradient. We might expect temperature to drive physiology, abundance, diversity, and metabolism. and I'm going to focus on biodiversity today. So early work in Iceland has shown a few different things. This is all research by Guy Woodward and Owen O'Gorman, who've been working in the Iceland part of this experiment for about a decade. And they found that invertebrate diversity seems to go up with a small amount of warming, and then it declines rapidly. Diatom abundance decreases with warming, and invertebrate body size increases with warming, among other things. But we wondered whether this was just an Iceland effect. Would it be different in different regions due to different regional species pools and different other things to do with biogeography? Because these sites are all fairly different. So some of them are in the full Arctic, for instance, Svalbard is in the Arctic tundra, and others of them are more in the subarctic and are actually quite forested. So here we have Disco Island in Greenland in the top left. So you can see it's quite tundra-like. In Svalbard at the very bottom left, we have very icy location, and in Kamchatka and Alaska on the right, they're much more forested. So you might expect there to be a different effect of temperature in these streams due to this surrounding differences in the terrestrial landscape. So as I said, we were looking at everything from gene expression all the way up to whole ecosystem processes. The part that I was working on was the food web. So looking at everything that lives in the stream, from diatoms to macroinvertebrates to fish if they were there. So this involved collecting hundreds of samples, identifying them, measuring them, to get species richness lists, which I'm going to talk about today, but also eventually to look at food web responses. So yeah, this is some diatoms, some pictures that Sarah, who's worked on this part of the project, has taken. So to jump straight into some results, it appears that richness does decline with warming. So importantly here on the x-axis, we have warming above the coldest ambient stream in each region. So we can compare the trends across the regions, which might have different air temperatures because they're at different latitudes. And we have richness on the y-axis, and this is the invertebrates. So overall, there seems to be a clear decline in richness with warming across all of the sites. So this is about 40 streams across the 5 locations. But it depends on the scale. If we start looking at this within each region, which is what I think we should do, you can see we are seeing declines in richness with warming in Svalbard in pink, in Disco Island in Greenland in blue, in Kamchatka and Russia in orange. But in Iceland, so that's the Hengel site in green and Alaska in yellow, we're actually getting almost stable or almost increasing richness with warming. So this highlights the importance of scale, because if we just look at the large scale, we see an overall decline, but this doesn't reflect what's happening in some of the locations, and the importance of biogeography, because there appears to be a different relationship in these different locations. So we asked what drives this variation. So we looked at various different things. So the chemistry of the stream, for instance, but also biogeography variables. And this is an ordination plot where each point on the plot represents a stream. And if they're close together, it means the streams are similar to each other in their community. And if they're far apart, it means they have very different communities. But what the important thing to note here is that the first axis separating these streams appears to be driven by biogeography. So the latitude of the sites, the size of the sites, whether they're isolated or not, and their NDVI, which is a normalized vegetation index, which essentially shows how green the site is, so how much terrestrial productivity there is. And the second axis is driven by stream temperature. So we looked at this a little bit further to see what the best model was to predict the effect, to predict species richness. And we found that the best model was a significant interaction between temperature and NDVI. So overall, it appears that warming causes the decline in richness, except in locations which have high NDVI. So except for in locations which are particularly green. So with lots of surrounding grassland and terrestrial productivity from forests, for instance. And we think this might be something to do with metabolism. So as streams warm up, the metabolic demands of invertebrates will go up. And if there's not enough resources for them to keep up with this rising metabolic demand, then we might lose abundance and richness. But if the surrounding terrestrial landscape is providing additional resources to the stream from terrestrial subsidies, then maybe they're able to keep up with these rising metabolic demands and we don't lose so much biodiversity. We see similar results for diatoms, but again, there is slight variation depending on the scale and biogeography. And here, richness was best explained by an interaction between temperature, NDVI, and biome area. And as you might expect, there was higher diversity in continental regions, so in Kamchatka and Alaska than on the islands. And NDVI again seemed to kind of modulate the effect of temperature. So particularly in Iceland, slowed down the declines of richness with warming. We also looked at this using beta diversity. So here we have the temperature difference between paired streams on the x-axis, and then how similar the stream communities are to each other on the y-axis. And as you would expect, streams which are more similar in temperature have more similar communities, and streams which are very different in temperature have very different communities due to different thermal tolerances. That's not surprising, but what is interesting is that this turnover rate, so the change in species between streams, happens at different rates, and it appears that there is higher turnover in locations with lower average NDVI. So this means that in locations which are much greener, the turnover in community structure or dissimilarity appears to be less. So again, somehow the terrestrial landscape is modulating the effects of warming in these streams. But importantly, this is just a snapshot of these communities. So these are from samples collected back in 2013, I think. We've since returned and sampled the streams again, and also expanded it to about 80 streams to try and disentangle the effects of temperature and biogeography. But also these streams have been heated for hundreds, if not thousands of years. So many of the species may have acclimatized to the stream temperatures. And this is not what's going to happen in the real world with climate change. So it leaves the question, how will communities change with rapid warming? And I'm sure many of you are familiar with this figure from the IPCC, which looks at predicted global temperature increases with various socio-economical scenarios over the next century or so. And we're predicting increases of between 2 to 7 degrees Celsius, depending on what scenario and where you are in the world. So this is much more rapid warming than we might get in our natural experiment in the Arctic. So to look at this, we've used experimental mesocosms. So these are some mesocosms that we have set up. There's some in Oxford and also in Silwood Park, where I did my postdoc. And this is a schematic showing the setup in the Silwood Park mesocosm facility. So we've set the mesocosms up like a 96-well plate. You can see in the drone shot there. And the schematic shows the treatments. So the blue mesocosms are those which are ambient, so they're not heated at all. And then the number shows the amount that the mesocosms are heated above ambient background temperatures. So we have a temperature gradient from 1 to 8 degrees above ambient at 1 degree increments. So this has given us a nice temperature gradient. So this is a figure produced by Danny Harris, who's a postdoc at Silwood. And this mesocosm facility is led by Emma Ransom and Guy Woodward. So you can see we've got a nice temperature gradient. This is the mean daily temperature over the year. So this has been warmed now for 2 years, from about 12 degrees annually in the ambient ponds to about 20 degrees. So we can see how this smaller temperature gradient, but more rapid warming will affect the communities. So we're looking at all sorts of things in these ponds, the same as the Arctic streams. My part, again, is focusing on the food webs, and I want to talk today about the invertebrates and the question, does warming favour the small? So there's a rule from biogeography called Bergmann's rule that predicts that as temperature increases, body size should decline because of surface area to volume ratio. So it benefits organisms to be smaller in warmer regions and larger in colder regions. And this example is of bears, so you get polar bears at higher latitudes and some bears, for instance, nearer the equator. And you can also see this rule within species. So for instance, moose are larger at the more northern parts of their range than the more southern parts. And there's plenty of evidence to suggest this rule exists in both endotherms and ectotherms. So the examples I have here are fish, phytoplankton and birds. From our own research in Greenland, we've seen this is just a quick example rather than showing too much data. So in a cold 2-degree stream, the communities are dominated by this Diptera larvae, Procesimulium, but just by warming the streams by 4 degrees Celsius, we have a change in community structure to these much more smaller bodied species, Simulium. However, we've seen the opposite in Iceland. So this is some work by Owen O'Gorman that shows that trout, which are the largest predators in these streams, are more abundant in the warmer sites. And actually the average body size of invertebrates increases with temperature. So it appears that there's something else going on here. And this is why it's important to look at what happens in different locations, to look at different context dependencies. So we've just started looking at this in these 96 mesocosms, looking at zooplankton. So as a PhD student, I spent hours on a microscope counting and measuring different zooplankton and macroinvertebrates. One of the first things I've done as a new PI is try to take away the step for my postdoc, especially when I'm asking them to look at 96 ponds. So I was lucky enough to receive funding to buy a flow cam, which is essentially a flow cytometer coupled to a microscope, where you can build libraries of different plankton and use machine learning. So the machine essentially identifies and measures and sorts the sample itself. And this is work that's being done by Dania, a postdoc in my lab. So she's been taking samples from these ponds in Silwood, running them through the flow cam. And this is some of the examples of the images she's produced from ambient ponds in blue to ponds heated 4 degrees above ambient in orange there. And it's produced an astonishing amount of data in a very short period of time, probably more than I've produced in my whole career, I think. And so these are some very rough and ready figures that we've just got this week from the flow cam, looking at copepods, nullply and adults across the temperature gradient from 2 sampling points in spring and autumn. And obviously there's lots of variation there. We need to look at this in a bit more detail, but it appears that body size does seem to decline with warming, particularly in spring in pink and particularly in the adults. But if we look at other organisms, we see different responses. So this is Carotella, a rotifer, and its body size appears to increase with warming in April, but then decreases with warming in October. So Dania tells me that we have a lot more data to add to the October samples. This may change this, but again, there seems to be more context dependency going on. So I don't think we can say that warming favouring the small is a universal rule. It depends on location, it depends on taxa, and it also appears to depend on season. And it might also depend on other stressors present in the system. So I want to move on now to talk about multiple stressor interactions. So what do I mean by multiple stressors? So I refer to them as any anthropogenic stressor. So this could be those associated with land use change, for instance, deforestation, urbanization, agriculture, all of the pollutants that are associated with this land use change, and human activity, so plastics, pharmaceuticals, biocides, fertilizer, sewage pollution. Also invasive species, so the example I have there is the red swamp crayfish, but also climate stressors, so all of the stresses associated with global climate change, including extreme events. And I think freshwaters are uniquely susceptible to multiple different stressors. Because they occupy the lowest point in landscapes, they can accumulate stresses occurring in the wider catchment area. So they'll be affected by stresses occurring within the freshwater system itself, for instance, invasive fish, exploitation of resources, including water abstraction, pollution from boating activity, habitat modification, such as the building of dams, but they'll also be affected by stresses occurring in the wider catchment area. So things like invasive riparian trees, which will alter the input of resources to the aquatic environment, At the wider catchment scale, land use could cause eutrophication or pesticide pollution. Habitat change in the wider catchment can alter flow rates and change the structure of the river network. And of course, all of the systems on Earth are now impacted by climate change and all of the associated stresses, including warming, heat waves, and droughts and other extreme events. So one of the ways that ecologists try and unravel the impacts of these multiple different stresses is by using Mesocosm experiments, similar to the one I just talked about at Silwood Park, where we look at the impact of stressor A and stressor B, both alone and together compared to a control in a fully factorial design. And normally these stresses are kind of switched on at the same time in a very simple scenario where we just have one stressor or no stressors or both stressors. And really what we're trying to ask with this is, can we predict the combined impacts of the stressors by understanding their independent effects? So can we just add up the effects of the two stressors to work out what happens when they occur at the same time? So in this example here, if we have a population of fish and we lose 3 individuals because of a heat wave, and we lose 2 individuals because of a pollution event, with an additive null model, you might expect mortality of 5 because 3 plus 2 equals 5. Another null model that's commonly used is the dominance null model. And this is where you just take into account the worst stressor because essentially you can't die twice. So in this case, that would be a mortality of 3. And then the final model is multiplicative, which would predict mortality of 4 with our example here. So we have a range of null model predictions based on our simple example where we have mortality of 30% with a heat wave, mortality of 20% with a pollution event, and our null model would predict when we get these two stresses occurring together, we have mortality of 30 to 50%. So we wanted to ask which null model is the best. So this is work by Olivia Morris, who's a PhD student, and with help from me and Charlie Lowen, who is a researcher at the University of Toronto, we've built a database of about 400 responses to multiple stressors, looking at various different stressor pairs from temperature and nutrients, and temperature and contamination, which have got an awful lot of response endpoints, through to stressors which have been less well studied, but still included in the database, and responses measured across lots of different taxonomic groups and organization levels. And if we jump straight into some results, we wanted to see which null model was better at predicting the combined effects of these different stressor pairs. So here we have T for temperature crossed with a few other different stressors, and the null model which was the best, so the frequency of that being the better null model. And in dark grey we have the dominance null model, which appears to be the best in most cases in about 50% of the database, but there doesn't seem to be a clear trend. So we were hoping that we'd see that the dominance null model is better when you're looking at a certain stressor pair or a certain response point, but it seems to be fairly random. The one trend we could find is that if the stresses are very asymmetric, so if one has an effect which is much larger than the second stressor when they occur independently, particularly if it has an effect which is more than 50% larger when they occur independently, then you can almost definitely assume that the dominance null model will be the best predictor. So you can disregard the effect of the stressor which has a smaller impact when it occurs alone, particularly if this larger stressor is habitat alteration, nutrients or contamination. But importantly, these null model predictions are often actually out, even when we look at these 3 different null models. So again, here with our simple scenario, we have a prediction of mortality of 30 to 50%. But our database showed that in 76% of cases, the observations fell outside of the null model range, with almost half of them being smaller than predicted by all null models. So back to our simple example here, in this case, we might get a mortality of only 25%. So this is less than what any of the null models predict. So somehow the stressors are interacting to mitigate one another's effect. And this is often known as an antagonistic interaction between stressors. And then at the other end of the scale, in about 30% of our observations, the impact was larger than predicted by any of the null models. So here in our simple scenario, again, we might have mortality of 60%, which is larger than predicted by any null model. So somehow the stressors have interacted to amplify one another's effect. And this is often known as a synergistic interaction between stressors. So it appears that we're commonly over and underestimating multiple stressor effects. And so many null models aren't really fit for purpose. So it was only in about a quarter of the database that the null model was able to predict what actually happened when multiple stressors interact. In almost half the database, the impacts of multiple stressors was less than we could predict based on any null model. And in the remaining 30-ish% of the database, the impact of the two stressors was larger than we predicted based on the null models. So we started thinking about why this might be. And one of the reasons perhaps might be that all of this data is based on these simple controlled experiments, which might be oversimplified and unrealistic scenarios. So as I said earlier, both the stressors, I'm calling them here stressor A and stressor B, are switched on at the same time and switched off at the same time, which clearly isn't how it works in the real world. In the real world, stressor magnitude varies over time. So the world is getting warmer, but this is accelerating in time. Extreme events are fluctuating in time, although increasing overall, they still come and go. That's why they're extreme events, such as wildfires. This is an example from my own work, looking at biological invasions in the Thames catchment, showing how the number of species that invade each year varies over time, with this accelerating from about 1930 onwards for both accidentally and intentionally introduced species in grey and black, respectively. The use of biocides is varying over time as new ones come and go and are banned. So we're getting fluctuations in herbicides, fungicides, insecticides, different pharmaceuticals, and also variation in forest cover, which is accelerating in loss over time. So we need to consider this temporal dynamics of stressors. We can't consider them to be consistent in most cases. They might be discrete and they might have fast or slow onset. They might have fast or slow dissipation. They could oscillate in time. They could be ramped. They could be stepwise. And we need to think about this whole temporal complexity to really understand how multiple stressors interact. And this is another couple of real world examples looking at nutrient pollution in an estuary in Florida. And you can see that nutrient pollution is much worse in some years than others. So it's oscillating in time, whereas global warming is a ramped continuous stressor. But what's important to note here is that this is very much from a human perspective. So another longer lived organism, such as a predatory fish, might also experience the nutrient pollution as an oscillating discrete stressor, so varying from year to year. But a species with a shorter generation time, such as a single-celled algae, if it occurs at that point I've indicated on the figure there, would experience this stressor as a ramped continuous stressor. So it's really important when we think about the temporal dynamics of stressors, we consider how they vary in time, but also how this will be perceived by the organism we're interested in. So I think we need to move beyond these simplistic scenarios of multiple stressor experiments to look at something a bit more like this. So this is just an example of a few of the treatments that we're working on. And this is work with James Orr, who's a postdoc in my lab, and Adriana, who's a PhD student. So we're going to be looking at multiple stressors that occur in the simple way in the top part of the figure there, and then staggering stressor effects. So what happens if stressor A comes before stressor B and vice versa, and what happens when they have different degrees of overlap. And we're able to do this using a unique Mesocosm experiment where we have 128 stream channels where we can manipulate stressor order and magnitude in this way. And what we really want to get to grips with as well is ecological memory. So not only are we going to be manipulating stressors within the experiment, we're going to be looking at this across the land use gradient. So does the historical exposure of the site affect its future response to stresses such as heat waves? So we'll be looking at this in sites which are quite heavily impacted by humans and more pristine sites to see whether this affects their community response. we're also interested in how it might vary with biogeography. So we're working with partners who actually designed this infrastructure in New Zealand, Ireland, and Germany. So that's something that we're working on at the moment. So that's a watch this space. And if you're interested to read more about the temporal dynamics of stressors, we've just had a paper come out in Trends in Ecology and Evolution. And if I've got time, I'm going to talk a little bit about a kind of proof of concept study that we did to look at this in South Africa. So here we were interested in the sequential effects of multiple stressors. So asking, does previous exposure to warm conditions alter how communities respond to further stress? So you would expect that it would, because vulnerable individuals or species might already be lost to the first stressor. And the remaining community may have acclimatized or adapted, so it will be more tolerant to the stress. So we looked at this in another geothermal stream setup. So we were just focusing on two streams here, one that's heated to about 15 degrees Celsius above ambient, and one which is ambient stream temperature, to just reflecting the surrounding air temperature. And we were interested in the microbial communities here. So we took sediment from the hot stream, put it in tubes, put it back in the hot stream, and the same in the ambient stream. Then we took sediment and translocated it between the streams, and half of these tubes were also applied with a pesticide. So we have a fully factorial design here where we have sediment that's left at the temperature that it was taken from, sediment that's left at the temperature it was taken from and treated with a pesticide, sediment that's been translocated to a different temperature, and sediment that's been translocated and treated with a pesticide. And these are provisional results just looking at bacterial copy numbers per gram of sediment, so essentially showing us the abundance of bacteria in each of these treatments. So the larger the bar, the more bacteria. And firstly, in streams, in sediment that came from the cold ambient streams, it appeared to be that there was little effect of the treatments. And fairly similar with sediment that came from the hot stream, although there was much more bacteria in these samples. But if we look at this in a slightly different way, so just looking at sediment origin and bacteria abundance, you can see that sediment that came from cold streams, adding the pesticide had very little effect, whereas sediment that came from warm streams, adding the pesticide increased the abundance of the bacteria. So the pesticide appeared to promote abundance, but only in a warmer world. So the actual historical exposure of the communities to temperature was much more important than the experimental temperature. So I think that's everything I wanted to cover. So I will leave that there. Thank you very much for everyone that's helped. There's a long list of names there of people that have contributed to this work, and to all of the funders and Imperial College, where much of this work was done when I was a postdoc there. Thank you very much.
Thank you, Michelle. That was fantastic. So we have time for questions. So if you have a question, either raise your hand or post it in the chat, or turn on your camera and make it clear and wave at me that you want to ask a question. So if anyone has a question, please go ahead. While we're waiting for questions to come in, I had a question about so the change in diversity in different temperature regimes. And it seemed that in certain streams, it got to a threshold whereby it increased initially and then declined rapidly past a certain threshold. Are we getting towards those kind of thresholds in our kind of UK rivers in response to climate change and also other multiple stressors? And is there anything we can do to mitigate those kind of impacts?
So yeah, in the Iceland site, I think that was the one you're talking about. Yeah, the biodiversity increases with initial warming. So it seems that small temperature increases can actually benefit some of these organisms. And that's probably more within the range of what we would predict with the IPCC predictions of temperature increases. So, you know, maximum of about 4 degrees Celsius over the next few decades. But I think it's important to remind ourselves that those streams have been heated for hundreds of thousands of years, and in the 4 other sites there was declines with warming, even over a couple of degrees of warming. So I think Iceland's a bit of a special case there, and in most cases there are declines of warming, and I think there definitely will be with rapid temperature increases in sites which aren't so used to it.
Sylvie, did you have a question? You turned your camera on.
I do, yeah, thank you. Really interesting, if a bit scary. I was just wondering, is there anything that we could learn from environments which have enormously fluctuating temperature ranges? So I don't know, I was sort of trying to work out in my head what that would be, but I don't know, something like a pond in the Gobi Desert, something that would experience, extraordinarily high temperature fluctuations between, for instance, daytime and nighttime.
Yeah, I would certainly think sites which have more larger temperature increases, like these pond sites that you've mentioned, would be better at dealing with warming, because the organisms in them are more used to temperature fluctuating on a daily basis or a seasonal basis, whereas sites which have a more consistent temperature over time, so perhaps more sites nearer the equator, you would expect the impact of warming to be much more severe, because they're not so used to any variation in temperature. Is much more consistent annually. But I don't know if anyone's used that thought process to make predictions about the impacts of warming. I suspect they probably have, but I don't know of it.
Thank you.
David.
Hi, Michelle. That was really cool. Lots of really good stuff. I don't know if this is a question more than a thought, but as you talked about the effects of multiple stressors, it sort of made me think about a lot of the work that's been done on aquatic toxicity and how organisms respond to multiple stressors, and how those outcomes are modeled and analyzed. And I think if you haven't looked in detail at some of that, because it's a bit of a niche field, there's actually a lot of quite useful tools for presenting interactions between different stressors, which might be useful. And related to that, there's a lot of work that's been done on how the order of presentation of stressors can affect the outcome on aquatic organisms. Also, there's, I think, quite a lot of thought that's been given to how similar those stresses might be on affecting the physiology of the recipient organisms. And obviously, if the physiological impacts are quite different, then often you get the effect of one stressor weakening the organisms and another kicking it when it's down. But really interestingly, A plus B doesn't always lead to the same outcome as B followed by A.
Yeah.
But
yeah.
I don't know if you looked at it in much detail. I can send you a few bits and pieces if you haven't. But I think there's some really interesting.
Yeah, there's some really interesting overlap, and it's interesting that you said that, because we recently last year we put a paper out about how there's not enough communication between ecologists and ecotox people. So there was a group of ecologists, which is what I would consider myself to be, and some ecotox people, including Robbie Stocks and Luke Demester, who might be some of the work you're referring to. And James Orr, who's now a postdoc in my lab, used a networking approach to show citation networks of how there's lots of people in terrestrial, marine, freshwater and ecotox systems working on similar questions about multiple stressors, but not reading each other's papers or communicating with each other enough. And so we often use different terminology and have made similar advances and called it different things. And so there's a paper we put out last year talking about how we should kind of unify multiple stressor research across these disciplines. And yeah, I'd be very interested if you've got some particular papers you think would be key that maybe I might have missed, because I know there's a lot out there.
I'll send you something.
Thank you.
Andrew Bladen, did you have a question?
Yeah, thanks. That was really interesting. I was just wondering, this might be a slightly naive question because I'm not a freshwater biologist, but the streams with the thermal springs, where you get the massive range of temperatures, are obviously really interesting for kind of comparisons. And you sort of showed that the greater the temperature difference, the more dissimilarity you got in the communities. But I was wondering whether you've done anything to kind of look at, presumably there are a few species within those streams that actually, do overlap and therefore are kind of able to tolerate quite a massive range of temperatures.
Mm-hmm.
Is there anything that we can learn from, I don't know, the traits of those species or the taxonomic groups that they come from or whatever, about, what affects this ability, either a sensitivity to temperature or a tolerance of it, and therefore kind of which species are likely to kind of persist into the future because they've got this innate tolerance?
Yeah, that's a really good question, and it's not something I've looked at so much in all of the sites, but I know a lot of work has been done on this in Iceland, because Guy Woodward and his lab, and Owen O'Gorman, who's now at the University of Essex, have been working on that site for about a decade. And they've looked at the traits of different invertebrate species and how they vary across the temperature gradient. And I think a lot of it's got to do with body size and whether they've got a terrestrial life stage. So if they're able to kind of move between the streams and select them. And for the trout in particular, they do very well in the warm. They come into the warm streams to feed, but they don't breed in them. So they can have the fact that they got access to these streams and can move between them means that they can benefit from the warmth when it suits them, and then move away to the colder streams when they need to. And I think that might be the case with some of the macroinvertebrates as well.
Okay, that's really interesting. So it's almost sort of a false tolerance in that case, because they can't complete the life cycle there.
Yeah, exactly. For the fish, most definitely. Yeah.
Yeah. Cool. Thank you.
Ash, you have a question?
Thanks, Michelle. I was wondering, obviously you showed quite a lot of, you showed the 5 different sites and lots of streams within those sites, and all of them were quite different. So you summarised at the site level, and even some sites in different places showed different trends. So how much do you think you can extrapolate? So if you knew the environmental conditions in one stream, could you roughly say how, could you make inferences about how that stream or the communities would react? And are there particular species that might be able to indicate how the community would react? So I guess sort of relate to what Andrew is saying in terms of certain species coming out, but maybe if you know that the large predator in the system will react a certain way, and then you know roughly what the community's like, so you can kind of infer those responses for other species in similar systems. Obviously you can't do it in different types of rivers, but thinking of just rivers in the same sort of thermal heated rivers, do you think you can draw inferences from that, or do you think there's just too much variation?
I think there is a lot of variation. I think you could draw some to do with traits, like we were just discussing. So looking at particular traits that might make the organisms more tolerant. It's interesting because the traits that make them more tolerant seem to differ between the sites as well. So I think there's a lot going on that it's hard to tease out. So I would be worried about making too many predictions, because like I started at the beginning of my talk, I think we need to look at the kind of local variation and everything that's going on to get a real understanding of the impacts of humans on environments. Because although these systems that we've worked on are all very pristine, if you were then to start trying to make inferences about how this will affect other freshwater systems that are impacted by other stressors, there'd be lots more interactions going on that would be hard to understand.
I had a question about the multiple stressor database. So you had some that were under predicting the null model and some that were over predicting the null model. Was it certain stressors that were predominantly over predicting and certain stressors that were predominantly under predicting, or was it mixed across the kind of different?
That's what we were hoping for, that we would be able to get some really clear results and say, always when these stressor pairs or this response endpoint, you can say you're going to over predict or under predict, but it's really, really, really variable. Even though the data set is for more than 400 observations, I think we need more to understand what's going on. And also we've only used mean effects there. So maybe looking at the kind of variability in the data would give us a better understanding. But no is the short answer. That would be really nice.
Does anyone else have another question? Fred.
Hey Tom, good to see you again. Hi Michelle, that was really interesting. I think I can't remember where it was. It might have been BES or SFS. What I remember, I think you presented at the start of the Ring of Fire project, so it's great to see. It's great to see all the results come out now. It's really interesting. I was wondering, I presume you would have done, but I'm just interested about oxygen in this whole story. So the variation between the sites is obviously really quite interesting. And I was wondering, did you have, when you looked at the sort of biogeography, did you have the altitude of the sites in there as a potential factor, and you know how that interacts with oxygen saturation at different altitudes and different temperatures, and how that might be driving some of the physiological responses you might see in these systems?
We did have it in there. We had so many biogeography variables in there, and we started removing them because they were correlated. And also when we went back to the sites in 2017, we actually directly measured oxygen over 48 hours in each stream to get ecosystem respiration and look at those sort of oxygen demands as well. So that's work being done by Mark Trimmer and Sarah Harpenschlager at the University, Queen Mary University. So I don't know the results, I'm afraid, but I think something interesting should come out of that.
That's great, thank you. Yeah, no, there's a really nice body of work by Wilco Verberg from Radboud, looks at this interplay between oxygen and temperature and how you get these closing windows. Yeah, that's really neat. Thank you very much.
The other thing we were concerned with is how pH can change across the temperature gradient as well, but luckily that's not a important consideration in any of our sites except for Svalbard. So luckily their temperature appears to be quite a master variable that's not correlated with any of these other things.
That's great, thank you.
Does anyone else have another question? Okay, well, that just leaves me to say thank you to Michelle for a fantastic talk, and thank you to everyone for joining us today. There is now a kind of 4-week break for the CCI seminar series, and we are back in about a month's time. But again, thank you very much, Michelle, for a really fantastic presentation.
Thank you for having me. I hope to visit in person one day.